In the realm of medical advancements, few areas have remained as stagnant as antivenom production. For over a century, the process has relied heavily on livestock, particularly horses, to create these life-saving treatments. However, with approximately 100,000 annual deaths from venomous snake bites worldwide, the need for more effective, accessible, and universal antivenom solutions has never been more pressing. This article explores the fascinating journey of antivenom development, from its 19th century origins to cutting-edge research that promises to revolutionize snake bite treatment. The traditional approach: horses and polyclonal antibodies. The concept behind antivenom production is remarkably similar to vaccine development. When a foreign substance, such as a toxin or virus, enters the body, the immune system produces antibodies to neutralize the threat. Scientists in the late 19th century ingeniously applied this principle to create antivenoms. The horse-based method: venom injection. Horses, chosen for their size and robust immune systems, are injected with small doses of snake venom. Antibody production. The horse's immune system responds by producing antibodies against various components of the venom. Blood collection. After a period of time, blood is drawn from the horse. Antibody isolation. The antibodies are isolated from the blood, purified, and processed into a treatment suitable for human use. This method produces what are known as polyclonal antibodies, a mixture of antibodies capable of binding to multiple targets within the venom. Limitations of traditional antivenoms. While this approach has saved countless lives, it comes with several significant drawbacks. Species-specific. Antibodies produced against one snake's venom are often ineffective against others, necessitating different antivenoms for various snake species. Uneven coverage. The horse's immune response may not produce antibodies against all venom components equally, potentially leaving some toxic elements unaddressed. Allergic reactions. As animal-derived products, these antivenoms can cause severe allergic reactions in some patients. Production challenges. The process is time-consuming, expensive, and relies on the availability and maintenance of suitable livestock. The need for innovation. Given these limitations, researchers have been working tirelessly to develop more effective antivenom solutions. The ideal antivenom would be effective against multiple snake species, targeted towards the most dangerous venom components, less likely to cause allergic reactions, easier and more cost-effective to produce. Enter the world of monoclonal antibodies and broadly neutralizing antibodies. Monoclonal antibodies, a targeted approach. Monoclonal antibodies represent a significant leap forward in antivenom technology. Unlike polyclonal antibodies, which are a mix of different antibodies, monoclonal antibodies are derived from a single cell line and target a specific antigen. Advantages of monoclonal antibodies. Specificity. They can be designed to target the most toxic components of venom with high precision. Consistency. Being produced from a single cell line, they offer more consistent results. Reduced side effects. As they're more targeted, there's less risk of unwanted immune responses. However, the specificity of monoclonal antibodies also presents a challenge when dealing with complex venoms containing multiple toxins. Broadly neutralizing antibodies, the game changer. The latest frontier in antivenom research involves broadly neutralizing antibodies. These remarkable antibodies can bind to and neutralize multiple related toxins, potentially offering protection against venoms from various snake species. The phage display technique. To identify these superantibodies, researchers employ a method called phage display. Gene collection. A vast library of human antibody genes is collected. Phage insertion. These genes are inserted into bacteriophages, viruses that infect bacteria. Antibody expression. The phages express antibody fragments on their surface. Toxin exposure. The phages are exposed to specific venom toxins. Selection. Phages with antibodies that bind strongly to the toxins are isolated. Gene retrieval. The genes for these successful antibodies are retrieved and used to produce full antibodies. Expanding the scope. The brilliance of this approach lies in its iterative nature. Researchers can repeat the process with similar toxins, gradually identifying antibodies that can neutralize an entire family of related toxins. A 2023 study published in Nature Communications exemplifies the potential of this approach. Out of over 700 candidates, researchers identified a single antibody capable of neutralizing alpha neurotoxins found in the venom of several deadly snake species, including king cobras and black mambas. The promise of universal antivenoms. The development of broadly neutralizing antibodies opens the door to creating universal antivenoms treatments effective against multiple snake species. This breakthrough could revolutionize snake bite treatment in several ways. Improved accessibility. A single universal antivenom could be widely distributed, especially benefiting remote areas where specific antivenoms might not be readily available. Faster treatment. With a universal option, treatment could begin immediately without needing to identify the specific snake species. Cost-effective production. Standardized production of a single antivenom could reduce costs compared to producing multiple species-specific treatments. Reduce side effects. Lab-produced antibodies can be engineered to minimize allergic reactions in humans. Challenges and future directions. While the future of antivenom looks promising, several challenges remain. Comprehensive coverage. Ensuring that broadly neutralizing antibodies can effectively neutralize all dangerous components across various venoms. Clinical trials. Extensive testing is needed to ensure the safety and efficacy of new antivenom treatments in humans. Production scaling. Developing methods to produce these advanced antivenoms at scale and at a cost that makes them accessible worldwide. Regulatory approval. Navigating the complex landscape of medical regulations across different countries. Distribution and storage. Ensuring that these new antivenoms can be effectively distributed and stored, especially in remote or resource-limited areas. The broader impact. The advancements in antivenom research have implications beyond snake bite treatment. Other venomous animals. Similar techniques could be applied to develop treatments for other venomous creatures like scorpions or spiders. Toxicology. The methods used to identify broadly neutralizing antibodies could be adapted to develop treatments for other types of poisoning. Immunology research. This work contributes to our broader understanding of how antibodies function and how to engineer them for specific purposes. Biodiversity conservation. Improved snakebite treatment could lead to better human-snake coexistence, potentially aiding conservation efforts. Conclusion. A new era in antivenom treatment. The evolution of antivenom from horse-based polyclonal antibodies to lab-produced broadly neutralizing antibodies represents a remarkable journey in medical science. It's a testament to human ingenuity and the power of persistent research. As we move closer to the reality of universal antivenoms, we stand on the brink of a new era in the treatment of snake bites. This progress promises to save countless lives, particularly in regions where venomous snakes pose a significant threat to human health. The story of antivenom development is more than just a tale of medical advancement. It's a reminder of how scientific progress can address long-standing challenges, turning the tide against threats that have plagued humanity for millennia. As research continues and these new treatments move closer to widespread use, we can look forward to a future where the fear of snake bites is greatly diminished and treatment is more accessible, effective, and safe for all. In the grand tapestry of medical history, the development of advanced antivenoms stands as a shining example of how continued scientific inquiry and innovation can lead to transformative solutions, even in fields that seem to have plateaued. It's a powerful reminder that in the face of persistent problems, human ingenuity and dedication can ultimately prevail, ushering in a new age of medical possibilities and hope for those at risk.